Well, welcome back, everybody. We're going to take a look at the first couple of pages of Chapter 15 on enzyme regulation today. And we're going to start by talking just a little bit about balance, because regulation is all about balance in metabolism and in biological reactions. There's a wonderful quote here by Jacques Monod, one of the leaders in the development of molecular biology and also regulation, in fact, in metabolism. There's a picture here of a Calder mobile. I don't know if you know who Alexander Calder is, but he's done many famous mobiles in his time, and they all represent balanced forces in one way or another. And if you've ever been to the East Wing of the National Gallery of Art in Washington, D.C., walked into the giant atrium there, there's a lovely, huge Calder mobile in red hanging from the ceiling. It's just enormous and it's beautiful. And so check that out sometime if you get a chance. But we're going to talk about balance in metabolism. And the essential questions for this chapter are: what are the properties of regulatory enzymes? And how do regulatory enzymes sense the momentary needs of cells? And what molecular mechanisms are used to regulate enzyme activity? And even though we're only going to cover the first couple of pages of this chapter for us this semester, we're actually going to cover information from all those three questions and talk about all of them. So let's take a look. Here are the parts of the chapter, and we're going to go up to the special focus. We're not going to cover the special focus at all, and we're not going to cover the particular enzyme that is the model of allosteric regulation, and you'll see what that is if you take a look at the chapter. But we're really going to cover the first couple of items here: factors that influence enzyme activity. What are the general features of allosteric regulation? Can allosteric regulation be explained by conformational changes in proteins? Answer to that is a big yes, as you can imagine. And what kinds of covalent modifications regulate the activity of enzymes? Already in this course, we've talked about covalent modification a number of times. And then finally, we'll ask, is the activity of some enzymes controlled by both allosteric regulation and covalent modification? And as I mentioned, there's a terrific example of that in this chapter. We're not going to go into detail about that particular enzyme, but we'll simply say the answer to that question is another big yes. Okay, so let's take a look. Here we go. What factors influence enzyme activity? Well, the availability of substrates and cofactors usually determines how fast a reaction goes. And as you can imagine, as product accumulates, the apparent rate of the enzyme reaction will decrease. And that's because the product of the reaction is starting to compete with the substrate for the active site. And as there's more and more product, it's going to bind to the active site and reduce the activity of the enzyme for the substrate itself. Second point here is that genetic regulation of enzyme synthesis and decay determines the amount of enzyme present at any moment. Genetic regulation is very important. We won't talk about it in this chapter particularly, but we'll come upon that later in regulation, in all the different pathways that we talk about. Enzyme activity can be modified allosterically. Allosteric is a great word. It means other site, and so allosteric behavior is action from another site on a protein or enzyme. And then enzyme activity can be regulated through covalent modification. We're definitely going to look at that. And we have to consider what are zymogens and isozymes and modulator proteins, and we'll talk about those briefly in this chapter. The first figure here, figure 15.1, shows you enzyme regulation by reversible covalent modification. And we've mentioned the action of protein kinases and protein phosphatases before, but so that you know, a protein kinase is an enzyme that puts a phosphate on a protein. Kinase means puts a phosphate on. So if it's a protein kinase, you're phosphorylating a protein, not a small molecule like acetate or glycerol or something like that. And then in turn, a protein phosphatase removes a phosphate from a protein. And so enzymes, being proteins, are phosphorylated by protein kinases and dephosphorylated by protein phosphatases. And almost always the substrate for the phosphorylation is ATP, although later in metabolism we're going to show you a case where a kinase, a protein kinase reaction occurs with the substrate PEP, phosphoenol pyruvate, and that's only been recently discovered, but it's a great story. So you get the idea here anyway. Covalent modification of the enzyme affects activity. And so the enzyme might be catalytically active when it's dephosphorylated and then catalytically inactive when it's phosphorylated. It could be the other way around. There's many enzymes that are activated by phosphorylation and then in turn inactivated by dephosphorylation. Here you see the essence of a zymogen. A zymogen is an inactive precursor of an enzyme. Zymo, of course, referring to enzymes, and gen referring to generation. So this is the generation of an enzyme by cleaving a protein precursor, if you will. And a great example of that, even though it's not an enzyme, is insulin. And you see here on the left of the figure a proinsulin molecule, which is longer than final mature insulin. And what happens in the reaction is that blue segment of the sequence is removed by proteases and leaves you with the familiar A and B chains of active insulin. And proinsulin is 86 residues, and you remove almost half of the sequence of the molecule to produce the A and B chains, 30 residues and 21 residues respectively. So quite a bit of that proinsulin is removed to make active insulin. Here's another case, chymotrypsinogen, which we discussed earlier in chapter 14. And the O-gen, of course, tells you that this is a precursor. And you can see active chymotrypsin down at the second and third rows of this figure. Chymotrypsinogen is the precursor or the inactive zymogen. And proteolytic cleavage of that long zymogen gives you a couple of smaller pieces, the pi chymotrypsin form, which is actually two peptides linked by a disulfide bridge. And then further cleavage gives you the alpha chymotrypsin, the active form of the enzyme, which really comes in three parts, essentially. There are lots of other zymogens that you're familiar with. Trypsinogen makes active trypsin, and procarboxypeptidase makes active carboxypeptidase. Proelastase makes active elastase, and pepsinogen makes active pepsin. You might note here that there are two ways to name a zymogen. You can either add OGEN to the end of the active enzyme's name, like trypsinogen, chymotrypsinogen, or you can insert pro before the name of the enzyme. So a procarboxypeptidase is a zymogen that produces carboxypeptidase by an appropriate proteolytic cleavage. And the choice of which of these to use is really up to the people that have studied and identified the zymogen proteins and the active forms. So there's no particular rhyme or reason to why trypsin's precursor is called trypsinogen instead of protrypsin, but you get the idea. There's an interesting figure here about blood clotting. The blood clotting pathway involves a whole series of proteolytic cleavages, so these are actually conversions of zymogen-like proteins into active forms. And we won't talk about this, but if you're ever interested, you can refer back to this figure and learn about the O-gens, if you will, in the blood clotting pathway. Blood clotting has to be very carefully regulated, and nature has evolved to give you a whole series of proteins that very delicately and carefully regulate blood clotting so that your blood doesn't clot up too much. And you may already know that even a normal individual with normal activity and no particular injuries or cuts and bleeding spots on them, everyone clots a little bit all day long. You make microclots in your circulation so that the blood doesn't just pour through your capillaries and so on and cause you to bleed internally. And if you take an anticoagulant in a high enough concentration, you can actually break up all those clots and hemorrhage internally, and that actually happens with the agent called warfarin, which we've talked about before and which will come up again. This figure shows you how isozymes are organized, and isozymes are interesting because they are similar but slightly different subunits of a given enzyme. So you may have an enzyme like lactate dehydrogenase, shown here, that comes in two forms. There's an A form and a B form, and it's tetrameric in its active state. And you can either have 4 A subunits in a tetramer, or 4 B subunits in a tetramer, or any of the three combinations in between that you see here. And interestingly, different tissues in your body use different combinations of these. The liver is almost entirely the A4 form, but it has a little bit of A3B and a little bit of A2B2 and so on. And you can see that brain is almost entirely AB3 with a little bit of A2B2, and the heart is almost exclusively the B4 form. So these are isozymes, all with slightly different activities, slightly different dependencies on other factors that can influence the activity, and all specialized to work with particular tissues in your body. Isozymes. So, what about allosteric regulation? What are the general features of what we call allosteric regulation? Allostery is action at another site, and enzymes situated at key steps in many metabolic pathways are modulated or controlled by what we call allosteric effectors. These effectors are usually produced someplace else in the pathway, and the effectors may be what we call feed-forward activators or feedback inhibitors. And I'm sure you've heard at least the feedback inhibitor term before, but we may not be familiar with feed-forward activation, so we'll show you what that looks like. And a key to allosteric regulation is that the kinetics of allosteric enzymes are typically sigmoid in their shape. They are S-shaped instead of the rectangular hyperbolic shape of typical Michaelis-Menten type enzymes. So we'll take a look at that. Here's what a sigmoid curve looks like compared to the hyperbolic plot. And you remember that we talked about how the Michaelis-Menten kinetics really show you a plot of V versus S that is a part of the hyperbola function that you probably learned about in high school mathematics. And if you look at the article by Ali Naqvi at the end of chapter 13, you can see the conversion of a hyperbola equation into the equation of Michaelis and Menten. So that's the hyperbolic plot there. And as a contrast to that, allosteric enzymes are typically prone to sigmoid kinetics, and you see a sigmoid curve here. It's a big lazy S curve, and that's where the word sigmoid comes from. It's an S-shaped curve. Now, we aren't really going to say anything about allosteric regulation beyond this point, except to say that there are two classic models that you might want to know about, and these could be topics for the MCATs, so I'll mention them briefly here. Three biochemists, three chemists, if you will, long ago, three molecular biologists actually, came up with what's called the MWC model, which refers to the first initials of their last names. Jacques Monod and Jeffries Wyman and Jean-Pierre Changeau came up with the MWC model, or the Monod-Wyman-Changeau model. And it's important because it imagined that allosteric proteins can exist in two states: the R state, which is colloquially called the relaxed state, and T, the taut state. Now, there's really nothing relaxed or taut about these particular conformations, but those terms were used to describe these proteins, and you should just remember that the classic Monod-Wyman-Changeau model refers to R and T states, and the names, the letters actually came from relaxed and taut. In this model, all the subunits of an oligomer have to be in the same state, and the T state predominates in the absence of substrate, and the substrate binds much tighter to R than to T, as it turns out. We won't talk about all the aspects of the Monod-Wyman-Changeau model, and we'll simply refer to its counterpart, a different model. We won't even look at that. The other model that's been proposed to explain allosteric regulation was first proposed by Dan Koshland and his colleagues Nemeth and Filmer, and that's called the KNF model. And it relies on the idea that ligand binding triggers a conformation change in a protein. Now, that isn't actually imagined to happen in the Monod-Wyman-Changeau case, and so it's quite different in its setup. And again, we're not going to talk about it in great detail, but we'll show you the contrast between these two. Here's the notable difference between Monod-Wyman-Changeau and Koshland-Nemeth-Filmer. In the MWC model, the different conformations of an allosteric enzyme have different affinities for the different ligands, and the concept of ligand-induced conformation change is ignored. On the other hand, the KNF model is based on ligand-induced conformation changes. And I'm sure that these two statements provoke some questions for you. We won't have to worry about these points too much. I don't think they're going to come up in the MCATs, but you may want to be familiar with the existence of these two models for allosteric regulation. So that's all we're going to cover in this chapter. We're going to move on to Chapter 17, the Introduction to Metabolism. So we'll see you there in just a minute.